Okay, thank you very much for coming for the Belowground interactions in the changing environment session. So this is the first one of the two sessions. The second session will be in the Evans main auditorium. So me and Omer colleague was a little bit off, organized the session, and we were thinking of bringing together microbiologists and plants. So it will be both in the same session. We have in this session 2 plant physiologists and 3 microbiologists.
Hi everyone, my name is Alon Rapaport, and I'm a master's student in Blind's Lab, the Weizmann Institute, right? Where we study ecophysiology of trees. And I want to share with you my project about carbon metabolites that are transferred between trees via mycorrhizal fungi. So as most of you know, mycorrhizal fungi have a symbiotic relationship with plants. Specifically in the forest, this mycorrhiza have the main part is the hyphae, which can connect to the roots of the trees with the direct physical link, and allow the exchange of nutrients and sugars between the trees and the fungi. But some of you maybe don't know that this mycorrhizal fungi can connect to several trees at once and allow the transfer, as previously showed by others, to allow the transfer of carbon between different trees in the forest. Now, as you know, carbon is part of any organic compound that there is. So we wanted to find out exactly in my project, how does this carbon, in what form of carbon is this carbon transferred? What are the specific metabolites that are transferred? Here you see an illustration of how this hyphae is going through the roots of the trees. So as I said, we're trying to locate the specific compounds or carbon metabolites that are transferred. This purpose, our main method were based on isotopic labeling, where we gave carbon dioxide, where the carbon is carbon-13, and we could locate it after labeling. So we gave the carbon dioxide in form of gas, 3, where it fixed it in the process of photosynthesis and turned it into different metabolites. Later, he transferred it to its roots. And later, as we know from other studies, it can be transferred to the fungi and also to neighboring trees. To neighboring trees, the forest. So we could collect the tissues from this different pathway and try to locate the exact metabolites that contain this carbon-13 that was transferred from the donor tree. So the first layout we used was a container, which contained the 3 compartments. The first one was, we called it the donor compartment, where we planted a pine sapling, covered it, and gave it a carbon dioxide, which is labeled. And neighboring to this compartment was a control compartment where we had the oak sapling, which was completely separated from the donor tree below ground with plastic, preventing the hyphae or the roots to penetrate through it and prevent the carbon transfer. And the next side of this compartment of the donor, we had the oak sapling, which was separated below ground with a mesh, preventing the roots to penetrate, but allowing the hyphae of the fungi to cross from the donor to the recipient and allow this carbon transfer. We know from other studies that shading of the recipient plants can enhance this carbon transfer. So we also wanted to examine the effect of shading of recipients trees on this carbon transfer. And here you see pictures of how it looked like. We had the middle compartment completely sealed, and some of the trees, as you see, are shaded. Here we see some some results of labeling of the different tissues of leaves and roots from the different compartments. So as you see in the control compartment, we see that the normal values of leaves and roots of unlabeled tissue is around -27. And if we look at the leaves of the donor tree, we already see labeling after 4 days where we sampled. We see that the carbon has got fixed in the leaves, later to be transferred to the roots of the donor tree. Later, we looked at the roots of the recipient tree, and we saw that after 10 days, we located some carbon that reached the donor, the shaded tree of oak. That this carbon was reaching from the donor tree. So what we did was collecting this tissue that contained the labeled carbon and try to send it to metabolomic analysis to try to identify these metabolites that contain the carbon-13. So we saw from initial results, we see that a lot of these compounds are known in literature as building blocks for phenol and flavonoids biosynthesis. And we know that phenol and flavonoids are can be involved in mycorrhizal formation. So just to sum things up, we saw that carbon was transferred from the pine to the shaded oak that are carbon starved, and also that this carbon is in form of phenol and flavonoids. So we identified some metabolites in the pine, the donor tree, and some in the recipient tree, but we're still missing the fungi part of this transfer, and this is because it's very hard for us to separate this hyphae from the soil. So we decided to look at the spore carps of this fungi, and to also hard to make this fungi appear, the spore carp, the fruiting body of the fungi. So we decided to collect spore carps in the forest. So we had two forests, the Charuvit and Varam forest, where we did labeling, again with carbon-13, where we labeled, you see the pine in the middle, we covered with plastic sheet, and we gave this carbon with this tree carbon-13, and then collected leaves, roots, and fungi spore carps adjacent to this tree. We collected Suillus and Tricholoma fungi species, and we know that these fungi species are known to be involved in carbon transfer from pine to oak. So here, see some results from, again, labeling of the different tissues. So we see already after 1 day that the leaf got carbon, labeled carbon inside of it. So it means that it fixed the carbon dioxide to itself. Later, we see that this carbon was transferred to the roots of the donor tree. And later we also located it in the spore carps adjacent to this tree, peaking at day 5. Overall, we have like half of the fungi spore carps that we collected had labeling inside. So, okay, really exciting. So we collected the tissue with the highest labeling, and we send it to metabolomic analysis. Here we see PCA showing the metabolic profile of the different tissues, and we see already separation between, first of all, the metabolic profile of roots and of fungi, and also of the fungi from different species. So this is why we decided to treat each fungi separately in this analysis. So luckily for us, we identified 35 different labeled compounds that were transferred to the roots of the donor tree. We had different compounds. Some of them were amino acids, some were carbohydrates, and again, some flavonoids. And in the adjacent spore carp, we also, of the Tricholoma, we identified 125 different compounds, again, a lot of amino acids and also carbohydrates. And when we look at it closely, we saw that 26 metabolites were common between the roots of the pine and the adjacent Tricholoma spore carp. If we look closely at these compounds, we again see a high portion of amino acids, but we also located some carbohydrates, amino acids, nucleosides, that were probably transferred directly from the tree to the spore carp, because we locate them in compartments. But we see also 99 compounds that are located in the tricholoma, but are not located in the root. This is probably because some of them were metabolized from these initial compounds that were transferred to them. If we look at the Suillus also, here we see that there are 15 compounds that are labeled compounds that were common between the tricholoma, the Suillus, and the root of the trees, indicating that these 15 compounds may be more generalistic and maybe appear in more species. Again, some carbohydrates, amino acids, and nucleosides. So to sum things up, we saw that in the containers, we saw carbon that moved from the pine to the carbon-starved oak, the shaded oak, form of phenols and flavonoids. In the forest, we saw that carbon moved from the tree to the adjacent spore carps. Some of them were amino acids that were moved directly. Some of them were other compounds that were probably biosynthesized from the fungi. Next, we will want to combine these two experiments together in the forest to labeling again with carbon-13 and try to trace also the metabolites that are transferred to adjacent tree in the forest. Hopefully, this could help us achieve the goal of finding out the ecological meaning of this carbon transfer. I want to thank my advisor, Dr. Tamir Klein, staff, and also my lovely wife for the support. Thank you.
Thank you very much for this great talk, Alon, really. Any questions, comments? Something? Yes, come on.
Thanks for a great talk. I wonder how fast the process occurs. It seems that in the container, you saw if there was an effect, it was immediate, and in the forest, it peaked and then went down. So is that what you expected before, or do you have some measurements that can?
First, we expect a peak and then decline because it gets diluted with carbon dioxide that is fixed after the labeling, which is not labeled. So we expect this dilution. We saw that in the spore carps, it took around 3 days already to the carbon to move to them. But we didn't check the root. We didn't see the carbon in the adjacent roots. So probably it takes more time in the forest. The scale is bigger and the distances are bigger. So it probably takes more time there. Yeah.
Anna?
Thank you. This was a wonderful talk and question is, how do you separate the hybrid? So how do you know that those relative to the high?
That's kind of a tricky aspect of this research. We try to. I don't really have a good answer for it, but I think it's hard to separate it.
What Rotem did.
Yeah, so Rotem did. Someone from our lab also looked at the DNA of the fungi, and when he extracted DNA from the roots, he separated the fungi DNA and the root DNA, and we actually saw that some of this carbon was transferred directly to the DNA of the fungi. Where we sequenced the fungi from the root, we saw that some of it contained heavy carbon. So we know it reaches the fungi part, but separating the metabolites from the roots.
Last question.
Thank you very much for the talk, very interesting. And this is not a question, I think it's an idea to use. Maybe you should check also what's happening with the saprophone. Rapaport survives also to there.
So I collected Rapaport in the forest, and we didn't see any labeling. So,
Thank you very much. Professor Ariel Novoplansky from our institute.
Thank you, everyone, especially to the first two speakers, which made a beautiful segue to my talk, because they were talking about exudate, root exudates, and communication via hyphae, etc. So this is a perfect place where we can ask what plants can learn from their environment, especially from other plants around them. When we talk about learning, we look at something simpler. Yes, we talk about a scenario in which we have a stimulus, and then we have some response. This is the first stage, and then when this and some memory, they memorize the response and the stimulus. Second time there is the same stimulus, there is an altered response. This is all. This is the schematic learning, and we are used to such schemes. In this case, you see children learning from remote learning in the 30s of the 20th century. You can guess why. This is not COVID. I leave it to you to think about. Let's think about the following situation. Okay, when we talk about plants, there is a certain plant standing out there, experience some stress. Could be drought, salt, or anything else in our system. And what about the neighbor? Is the neighbor oblivious to this, or can it get any information from the neighbor and do something about it before the stress hits it? Okay, so we ask specifically two questions. One, can neighboring plants communicate stress? And the other question is, can unstressed plants, which are the neighbors, relay the stress cues to yet additional neighbors? So think now the experiment itself, it's as simple as it can get, okay? A 5-year-old might, with good hands, can do this. You take a little pea seedling, you remove the root, and then you let it regenerate. You choose the ones with 2 roots, which are more or less similar in size. Then you plant them in such a shared scenario. They can share root exudates and other things, right? They can also share volatiles, so they can communicate. Right? And then you can induce some stress on one of the roots of one of the plants, and you can ask whether it will respond. Obviously, it should, right? But would the neighbor respond as well? Stress that it did not experience. Okay. To test for relay communication, all you need to do is to add some more neighbors. So this is a one-dimension type of a scenario, right? Very simplified. This is how it looks in the lab. You can change and manipulate the content of the cubes. You can induce all kinds of damages and stresses. And for the case of drought, you can measure stomata aperture, which is a very, very rapid and kind of easy to measure indication for stress in plants, especially drought or osmotic stress, right? So you measure the stomata opening, aperture, and this is what you get under benign conditions when you only induce some, you simply pump out and in back the water. So this is the norm in our environment for open stomata, right? But when you induce drought on one of the roots of one of the plants, after 15 minutes, both the induced plant and the 2 neighbors close the stomata, see the blue line, and after 1 hour, all of them close. 5 neighbors down the road, right? So this is a very rapid communication of stress cues. Okay, why to eavesdrop is very easy to understand if there is a very nice correlation between what happens to your neighbor now and what might happen to you next, right? It's easy to imagine. Why to be leaky? Why to provide information to neighbors is more tricky. but it can be easy to understand if you think about it. Many plants are rather large. So the information they share in the roots or trails might be with their own tissues, members of the clone, etc. Right? Some and many of the species we have on the slopes here are also very segmented. They are sectorial, which means that in order to communicate between different organs of the same plant, you have to communicate through the outside, right? External communication means that it might be exudates, which are exuded in one part of the plant and being received by others. Is it roots or shoots, right? It could have been either. So this is the system you have seen so far. This is shared, right? So each one is sharing its root system with the next neighbor. And all you need to do in order to test for this is to add another side to this experiment with unshared systems, right? As you can see here. Each and every one can only share volatiles from the shoots, but not the roots. And this is the thing, this is the result you have seen already with the shared, and this is the unshared. So this is clear-cut demonstration that the communication is in the roots. We don't know if it's really some metabolites which are volatile or not. We'll see it in a moment. Okay? So what we have so far is that some information is being shared. The information is about anticipated stress, supposedly, and the means is root queuing and relay queuing. Not only the next neighbor, but this neighbor who never was exposed to actual stress still relays the information, which is not trivial. Is it cry wolf situation? Is it just the stomata and then the stomata close and then they open and nothing happened? So is there any adaptive implications for receiving such information from the neighbors? So here comes the next experiment, which was done with buffalo grass, just published. The same situation, but simplified. We only have one immediate neighbor and one relay neighbor. This is the cheapest way to do it, in order to test for both communication and relay communication. And then you either induce for drought or you have control, benign controls. And you do it for 1 or 2 or 5 days. This is how you get some pot systems like this with the 3 plants, they induce and 2 target neighbors. And then you either induce drought in one of the roots, one of the plants, right, as before, or not. And then you test both systems under drought or no drought, right? And this is very important in order not only study the effect of the adaptive effect of such perception, but also the possible cost. You are not pressed later on. So this is surviving of the targets into drought. You can see an advantage to plants that were communicated by stress signals in the roots. And this is the, you can see 14 to 28 in the T1 and T2, advantage in survival into drought. Okay, so early perception of drought signals, in this case, we should call it cues, makes you more adaptive to future drought. And this, I'm just going to show you one example. I have to admit it's pretty extreme. After 30 days in the greenhouse here in Sde in the summer, buffalo grass set would look like this, totally dead and dry, brittle in fact. And if there was communication, some of them look like this, not all of them. This is a biased example. And what about performance? As can be expected under drought, which immediately came after, so there was not enough time to show differential growth. But there was some cost. Under benign conditions, the cued plant showed some cost. Okay, so if you're being communicated for incoming stress and you respond, somehow, physiologically, you may pay a cost for this if that drought will not come. The immediate suspect was this molecule. Some people like molecules here. Guess what it is? You should know, right? This is ABA. good old ABA, abscisic acid, which is being produced in higher concentration in drought plants, osmotic stress, and all kinds of other stresses, and also being exuded out of the roots under such scenarios. Okay, so this was the immediate suspect. The way to first give a first stab at testing it was to run the very same type of experiments, but putting one mutant on the way, on the chain. So this would screw up the communication, right? If ABA is at all involved, right? And this is exactly what we did. And you see that the induced plant, the two immediate neighbors up to the mutant. This is a wilted mutant which produces only 10% of ABA as normal, as the wild type. Were communicated well, closing their stomata under if the induced plant is being pressed, but hitting the mutant, there was very little response and the stomata kept open. This is in peas, but also the next in line. So the communication did not happen as well. We did it again with fluoridin, which is really messing around the biosynthesis of ABA in the plant, and with a very, very, very similar result. Surprisingly similar. So, and then when you measure metabolomically the amount of ABA in the rhizosphere, you get much higher concentrations of ABA, not only in the roots of the stress plant, but also in the next neighbor. So ABA is elevated. This is not proving that this is the vector, but this is just to support that ABA is involved. Right? When you run this very same experiment, the adaptive experiments that we did, that you have seen before with buffalo grass on peas, but this time not using drought, but using ABA, elevated concentration of ABA, the first stages of the induction here in red, you get very, very similar results. Okay, much greater survival time into drought. The benign controls are not here because they were 100% throughout. And there was even some performance implications with increased performance under drought, yes, with communication of the Q, with the communication of ABA queuing, and a cost, okay, under none. So what did we have so far? We have direct communication of stress cues. We have relay communication of stressful cues. It is all in the roots, between roots. So this is interplant root communication. And there is some adaptive implications. And ABA is at least involved, right? So we've seen that plant can learn. This is not the first time people have demonstrated that plant can learn. This is a nice way to show it in roots. And for the first time, I think that relay communication is involved, not only communication. Network communication could be with variable topologies. So should not necessarily be involved just fractional 1D dimension kind of lines of communication as done in my experiments. It could be much more complex, at least when you talk about real populations, real communities, right? So you may think about all kinds of communications. For instance, complications which are tremendously interesting for us. What would be the case in which your neighbor is not your kind? Maybe there is a neighbor which is xeric, more xeric than you are. When this neighbor exudes ABA or anything else, it would be more reliable. You have to listen to it more carefully. Be maybe even more hysterical about the situation, right? And vice versa. And this is just a segue to something I don't have the time to show you now. This also calls for further investigation of the cost of queuing of all kinds of information when the information doesn't match the future. So can plants unlearn or relearn or learn again some situations, and to what extent can they do it with certain costs? This is a whole another enchilada. I want to thank the group involved in this project and the funding sources. And thank you very much. And welcome any questions you might have.
Great talk. Great talk. We have time for a few questions. So, Dror.
So ABA is interspecies affecting different species, right? With can you see a plant?
It's a ubiquitous thing,
Yeah,
right? You have
yeah.
it even in fungi.
Right. So can one plant exude it in high levels to make its enemies suffer from closing the stomata?
That's a very interesting thing. Always when you deal with information, the question of cheating comes up, right? This is something I did very many years ago. I really went on and cheated plants to produce more crop, yields in crops, right? By giving them the wrong signal, right? So yes, there is such a possibility, but it's kind of blocked a little bit here. If your other roots would sense this cheating, right, high elevation of, let's say, ABA, you should be able to do much complicated things. You should be knowing that this is you, okay, providing the information, right? You know the fable about this old guy sitting below a tree in a village, and he was trying to sleep. And children came and harassed him. Say, guys, in the middle of the village, they give watermelons. And all the kids ran away and he slept over it. Then he woke up 5 minutes later and said, they are giving away watermelons, I have to run. So there is a danger when you try to cheat, you have to tag it with some information saying that it's you. You are in fact cheating. It's not very easy, but it's easier to think about situations in which let's say the high elevation of high levels of certain cues would be matched with the information about the type of neighbor giving it on. So if you know, for instance, if a buffalo is responding to the neighbor, which is Bermuda grass, and it knows that it is from Bermuda grass, it knows that it's coming from a xeric, and then the match, the tagging could be important. So it's not out of the question, but it's very hard to do.
What is the range of this communication? Does it have a finite attenuation, or maybe it's definite? Triggering processes locally.
Great question, very important question, not easy to answer question, right? Because we have shown in the lab, very easy setup, not easy to do, but think about 5 down, right? I would bet it could be even 10. Who knows, right? Let's think about it evolutionarily. What we need to have the good, tight enough, sufficiently tight correlation between what happens to the remote neighbor and what I'm going to experience in the future, right? So evolutionarily, natural selection should work so that it should be tight enough. Now, the level of tightness, strength of tightness is expected, at least theoretically, to be very different in different environments, different type of patchiness, right? Different taxa, et cetera, et cetera. So this would be under, it's not trivial. Not because it's not trivial for us to study, but the results for one set would be expected to be very different from another. But yes, it could be meters. Who knows, right? Or a few centimeters.
So beautiful work. I wondered, you excluded the fact they communicate through the shoot, basically not through the root, because there was no phenotype in this particular system. I wondered whether it could be about the fact that volatiles do not reach a threshold to activate a response. So what if you use a headspace which limits the loss of volatiles? Do they still don't have a phenotype?
Yes. That's really, really problematic. I know maybe you have done it before. When you put all kinds of covers and domes, especially in greenhouse situation or real light, high light situation, you get into trouble with radiation and heat. But put aside the technical problematics, yes, it is possible that some volatility communication happened here, but at least according to our lines, it might be negligible, at least in the plants we studied. I'm not saying that there is no, in fact, in the literature, you can find all kinds of evidence for volatile communication under drought and other things like salicylic acid, all kinds of metals, etc., etc. There is a long list, and reviewers are keeping mentioning it to us every time. Yes, and for good reason. But at least in our system, the way we do it, we even, in some cases, did the wrong thing and put systems near together. So it would mess up the results because we work in groups, in blocks, sorry. So it should, one treatment should mess up the other, and it never happened. So I'm not saying that it's not there, and it could be volatilic from the roots. Who knows? Right?
Last question, Kat.
A short one to follow up on this question. You think if there is drought stress that xeric one will announce there is stress because it will release it when it gets dry, and I assume that the mesic one will first experience.
Exactly.
Yeah.
Exactly. In fact, we have the result.
Okay.
I wasn't sure if I could, and I was very correct about stopping here. But we have some results, and they look messy because I didn't prepare it for the seminar. But here it is. When you put buffalo and Bermuda together, they differentially respond to each other. So, for instance, here they induced this buffalo, and the neighbors are either buffalo in the leftmost, this one. All of them are buffalo, like in the first experiment I've shown you. But here, the other neighbors, the T1 and T2, are Bermuda grass. And you can see now the yellow is the inverse log of, just to make it equal, of the dry versus benign. You can see that the shape of the golden line is very different. So they take, let's say, I'll say it in words, buffalo takes Bermuda Q8 much more seriously. It could be because of higher levels. We didn't do metabolomics, or this is the zero guy talking now. So this is going to be published very soon.
So thank you very much. Our next speaker is Professor Dror Minz from the ARO Volcanic Center.
Okay, so in my talk, I will take you from microbial ecology into more functions and probably even positive things to do to Earth eventually. The study is all based on the fact that just from our study by my office at that time, postdoc and a PhD and a postdoc in my lab, looked at the roots of plants and the bacterial communities that form a biofilm on it. So basically, for many years we are really excited about this thick biofilm of very active bacteria cover the root, and in a way they communicate with the roots in one side and the environment in the other side. So everything that exudates from the root to the soil or to the rhizosphere will have to go through this community, and everything that will move from the soil, including nutrients, but also pathogens, will all have to go through this very thick, in red, the biofilm of bacteria. There are orders of magnitude more bacteria on the root than root cells, very common to animals in a way. And the bacteria are extremely active metabolically, and you can see this is a fish, fluorescent in situ hybridization. So the stronger the signal represents much more active cells, because they will have more ribosomes to be labeled. And depending on the location of the root, you have more or less, but in any case, these are bacteria that are very intimately attached to the roots. If we're talking about what rhizosphere means, so these roots were shaken and then washed and then vortexed again. And these are the bacteria that will stick no matter what to the root. So they are really symbionts of the root. We wanted to, and of course, these are not one species. These are very diverse communities. And we wanted to see what are the types of interaction between them and the plant. And the first thing we checked is the community composition, and that's really communicates very well with the first talk today. So every different plant will have completely different community composition in the same exact system. So the same soil, the same pots, the same growth chambers, the same water, same student, and you see completely different communities, and actually 100% will be the total community, and you can see, for example, that the cereals will have dominance of one group. I don't look at the names, just ignore the names. You see high dominance of certain group of bacteria, and on the other option would be high diversity. So high dominance, low diversity, high diversity in the cucumber, for example, and this is all, those arrows are filled with hundreds of different species that are below 3% relative abundance, so that they don't deserve going into the figure. It really is an enormous diversity. So keep this figure in mind, and then we took the cucumber and the wheat and went on and tried to compare the composition, the metagenomic and metatranscriptomics of this community. So what are the genes that each community have on the different root system, and what genes are actually expressed in the different communities, the different roots. And okay, so the main question was that I'm going to ask here is, are genes expressed differently in the root microbiomes of different plants? Remember, it's in exactly the same conditions at the same time. Okay? So what we found out when we looked at the genes, the functional genes, and this is a tertiary plot that takes me too much time to explain, but basically what you can see is that in the root, in the soil, in the bulk soil of those pots, we see a community that has those genes. So approximately 400 genes that are, if you are a soil organism and not a root organism, you have to have this toolbox with you. However, in the root systems, we have 460 different functions that are basically, almost all of them are shared by the community in the two different plants. Okay, so these are approximately 37% of the functions are either overexpressed in roots or underexpressed in the root, represented, so not expressed in the soil. And the conclusion from here is that we have toolbox that bacteria should have when they are colonizing roots. In order to colonize the root, you have to have those genes. And regardless of what plant you are, and you remember that the cucumber in this side and the wheat in this side have completely different community, but we have functional redundancy. And if you really want to be on the cucumber, on the wheat, or on pine tree, or on eucalyptus or sequoia, you probably have to have those genes. What are these genes? So we did a lot of analysis, which I cannot really go into. I'll give you just a very simple way to remember it. In order to be a root community, you have to be able to sense the root, or chemotactic. You have to be able to move to the root, and remember that the root continuously moves. Okay, so when the plant grows, the root, at least the root tip and the most active part of the root is continuously moving, so you have to chase the root. You have to be able to communicate, so we have pure cone sensing. You have to be able to release compounds and enzymes and so on also to the environment. And you have to be able to eat the carbon compounds that the plant provides you, either by exudation, but also by degrading the cell wall. And the most interesting for us now is the denitrification or the simulatory nitrate reduction, because when we looked at the expression of the genes, this is a really exciting finding. Don't worry, I won't go into details, but these are the genes that are expressed in a high level, in green, in the microbiome of the cucumber, and in yellow, in a much higher level in the communities on the roots of the wheat. And there are so many things that we still don't understand, and we probably won't never understand, but we can, by looking at the green enzymes of import and degradation of organic matter, we can understand that probably these are the kind of organic matter that the plants, the cucumber plants have or provide, while arabinase, for example, is almost surely compound that is in high level in the exudates of the wheat and not in the exudates of the cucumber. Otherwise, why would the wheat community, not only one organism, many different organisms, this is metatranscriptome. This is a very strong signal. And silos, for example, is definitely in higher level in the environment, in the root system of the cucumber, and not in the wheat. But this is not the important thing. The important thing is that these genes, the denatrification, but also this set of genes are strongly suggesting that the community, once again, the total community, the diverse community, is expressing lack of oxygen, okay, in the environment. While the community in the cucumber root is basically screaming that they have too much oxygen, or at least not lack of oxygen. They even express catalases. So the community in the roots are telling us about the conditions in the root system, which is, I think, a neat idea, but not only neat. If I conclude now, plant microbiome is host specific. The plant-associated bacteria have specific toolbox of genes, but from that toolbox, they express differently in the different root systems, whatever fits them. And definitely, they will express differently in the day, than in the night, and in the winter, and in the summer. That's definitely extra issues to discuss in the future as well. And we can learn about the environment. So if we can we do something about it? Can we do anything good with the data that we got in ecological focus? And I want to focus again on the denatrification and zoom in on the genes. You can see that all of these genes are expressed in high level in the wheat system, in the community in the wheat system. What is missing is the last gene, NOS-Z, that is responsible for the enzyme that reduces nitrous oxide to dinitrogen, and nitrous oxide in denatrification, nitrous oxide is really bad. It's the villain in the environment because it's both extremely potent greenhouse gas. It's even calculated to be 300 times worse than CO2 as a greenhouse gas per molecule, per 100 years. Everybody ignores it more or less because there's a lot less of it in the atmosphere, but if you do the calculation, it's a big issue. But it's also, in addition to more than CO2, it's also a stratospheric ozone depleting gas. So we want to get rid of it as much as possible. And the EPA in the US calculated that approximately 3/4 of the anthropogenic nitrous oxide is due to agricultural soil management. And we hypothesize that it's it makes a lot of sense because if at least in cereals, all the genes to produce nitrous oxide are very highly expressed, and those to reduce it and get rid of it are not so much. That makes a lot of sense when you think about large wheat fields or corn fields and probably rice as well. So at that time, we asked ourselves, what can we do to minimize emission? Microbiologically, not chemically. There are means for, I don't know, all kinds of chemical means to reduce the amount of emission, but I'm not going to talk about it. One of the options is to just kill all of these organisms that produce nitrous oxide. Okay? However, this makes no sense because the denitrifiers are all over the tree of life. Almost every branch in the tree of life have denitrifying bacteria, and not only bacteria. And if you kill 99.99% in soil that we have 10 to the 5 different species per gram, what does it make? What does it do if you kill 99.99? Someone will come up and take over and do the same story over again. So killing them all will not work. And the next thing is to consume nitrous oxide before it's emitted from the soil. And that's a study of Alon Rapaport, that was at that time postdoc in my lab, and she's now in Weizmann Institute. And she wanted to find a bacteria that will actually fight the nitrous oxide or reduce it to dinitrogen. So you can see those bottles here. How much time do I have? Huh? 5. 55, so you want a type of questions. She isolated a lot of them. I won't go into the details how to do that, but microbiologists know how to do things like that. And and I'll show you 3 different bacteria that when they are in those closed bottles with no oxygen, but with a lot of nitrous oxide available as an electron acceptor, terminal electron acceptor, and it's quite good one in a way. You see the removal of the nitrous oxide from the atmosphere of those bottles. And that's the bacterial growth. And you won't see a different figure if it was oxygen. They enjoy the nitrous oxide, they reduce it, they get rid of their electrons so they can grow on the organic matter that we provided them. We did genome analysis. They all have NOS-Z, of course, because NOS-Z is the only enzyme known so far to reduce nitrous oxide on Earth. Maybe tomorrow someone will find another one, but so far with all the metagenomes and stuff, we see NOS-Z, all of them have it. Some will have all the other enzymes, some will have some of that. It's not important. They have the NOS-Z. So then she moved into a probiotic strategy where she took soil, healthy soil with all the 10 to the 10 cells per gram bacteria, natural bacteria, with all the enormous diversity, and then added her bacteria. This is how they look under the microscope. Added the bacteria to the soil, grew wheat in those pots, put them in a closed jar, and basically see some of the bacteria disappeared, some survived, and then she measured the nitrous oxide emission. And we did it in 2 completely different conditions. One of them is in ambient atmosphere that includes some oxygen, and bacteria will not do denitrification if they have oxygen, so this is not promoting denitrification, but she also removed the oxygen using nitrogen. It's not the nitrogen in the experiment, it's just nitrogen to flush the oxygen out. So this is anoxic conditions, and these are ambient conditions. And if we look at the nitrogen flushed, so the no oxygen in those jars, you see the syringe here sucking the gas to the GC, and you can see the control. This is the control in that condition when you don't have oxygen or almost no oxygen. We have very high level of emission, so it's 100 ppm in those jars. In normal atmosphere, it's about 310 ppb. Okay, so it's a really high level of emission, and this bacteria caused the plants to emit even more. This one, for example, decreased the emission by approximately 40%. And this is the number of pots. So I don't think anyone did an experiment with 81 different pots, and it's really very solid statistically. So this is significant indeed. So this is when there's no oxygen and the high level of denitrification, and therefore nitrous oxide emission. In an ambient condition, we have even in control a lot less. So instead of 100 ppm, a little more than 30 ppm, 66 different replicates. These bacteria removed more than 50% of the emission. Okay, remember this is a live plant in a very active soil, and the bacteria the bacteria she added is a minority of the community, but still they managed to do that. So the conclusion is that with microbial ecology, we detected the problem in the community, and we can actually use microbial tools to try to fix it. And we're now going into a big experiment. We look into different soils in Israel, some that will emit more, some that will emit less. And we're trying to use those bacteria in real life, more real than a pot with a plant in a greenhouse. And I don't think we will get 50-60% emission, but even if I manage to remove 2 or 3% of emission, I think I did my job and I can retire. I'm open for questions. Ilana, we can do it on coffee. Come on.
Okay, it's possible, okay, I don't know, it might be a tricky question. So if you think on nitric oxide for a second, not from the planet perspective, but I understand that from the bacterial community perspective, it actually has very important functions in the dispersal of certain communities. So the question is, does it come with a cost?
We're talking about nitrous oxide, N2O.
I know.
Okay.
No, I spoke on nitric oxide in the back. I'm just saying, when you look at the species, there is a community which does a certain function which is very bad for the earth, but it might be beneficial for the bacterial community which is on the road.
Okay.
So when you see how the microbiota change, you get a stable, diverse community when you introduce this bacteria, or you lose a lot of the diversity and functions of the bacterial community while improving the nitric oxide.
So about stable, I cannot say anything because we do it in one time point and we didn't do a time span, but there is a major shift in the community for sure, because when we looked with real-time PCR, we saw these bacteria that we added disappear in the soil with time. After a week, you could almost see nothing, but appear in the root system because they were isolated from the root system. From ecological point of view, we deliberately isolated bacteria that are root organisms. And they get up to 3-4% in the community. Definitely someone was pushed out. Okay, so they became quite dominant, right? And someone paid the price. I don't know if it's good or bad for the community. In the plant system, we didn't see it. The plants didn't show any.
Thank you very much again.
Thank you.
The question is edited with Professor.
בסוף הזה, פרופסור שתלמד.
Okay, perfect. So thanks to the organizers. It's always great to be in Sde Boker, and although there's a bit of rhizosphere in my lecture, it's not exactly the topic of my talk, which is antimicrobial resistance along anthropogenic environmental interfaces between ecological barriers and unseen. And when we talk about antibiotic or antimicrobial resistance, most people generally like to think about hospitals and clinics. And obviously, we know that overuse is causing a significant problem where bacteria are becoming more and more resistant, not just resistant to one antibiotic, but multi-drug resistant. But over the past 10, 20 years or so, there's this understanding that we have to look at antimicrobial resistance from an environmental perspective. And there's even a very good conference series that's been going on for maybe 10 years, talking about the environmental dimensions of antibiotic resistance or antimicrobial resistance. And one thing that I won't go into is the realization that antibiotic resistance in genes is actually something that's environmental. So these genes have been around for millions of years, long before Fleming first discovered penicillin. And that's a topic in itself. But the other is the fact that we are creating a tremendous amount of antibiotic resistance from hotspots, so what we can call anthropogenic hotspots, that are being transformed or transferred into the environment. Two of the largest are wastewater treatment facilities and animal husbandry. And of course, in animal husbandry, we're using a tremendous amount of antibiotics and obviously selecting for resistant bacteria, and these are being transferred through fertilization and other mechanisms into the environment. And obviously, we're not only talking about patients, but wastewater, which is going from the community, from the hospitals into the environment. And of course, in Israel, like some other water-stressed environments, this wastewater is being used for irrigation. So when I started at the Volcani Center about 12 years ago, Since treated wastewater irrigation is something that's very, very well studied, the question was, are we actually facilitating an increase in antimicrobial or antibiotic resistance by irrigating with treated wastewater? And basically the scenario is where in countries that are less water stressed, you generally see this type of pathway. Here we're looking at this type of pathway. And then there are a number of questions that we can ask, which are, what is the scope of antibiotic resistant bacteria and associated resistance genes in wastewater? And it's very, very important to realize that bacteria themselves aren't the only culprit, because we know that bacteria have the capacity to transfer genes through horizontal gene transfer, and many of these antibiotic resistant genes are located on plasmids. We have to look at the resistance genes as contaminants of emerging concern, not just bacteria. The second question is, do these elements persist in irrigated soils and crops? So it's not enough that have these transfer pathways. The question is, what's happening here? And a question that's really fascinated me over the past few years is, how do we assess the risk? We know that we can look at problems from the perspective of saying, yes, we have this there, but the real question is there really a risk? How much are these pathways really contributing to the global scope of antimicrobial resistance? So I'm going to touch on a number of experiments that have been done and a number of studies that have been done in the lab. The first was by Roberto Marano, who was a PhD student, and he used a solely culture-independent platform where we targeted a number of different antibiotic resistance genes, and we really selected them based on specific criteria that I won't go into here. They're kind of listed. Along with the 16S ribosomal RNA gene, which is, of course, a proxy for the total bacteria abundance. And then we can not only look at the total abundance of these genes by quantitative PCR, but also the relative abundance by normalizing to the 16S ribosomal RNA gene. And what we did, we had a very, very good connection with the Ministry of Health field inspectors, and we had access to several different wastewater treatment plants. So these are the blue triangles over here. And also to fields that are irrigated with the wastewater from these plants, which are the green circles. And in a number of cases, not always possible, but we did have access to specific sites that were irrigated in tandem with treated wastewater and freshwater. And then it's a lot easier to do comparative analyses. I won't go into all of the details, but first of all, when we look at the abundance of these genes in water samples, and these are in copies per ml, we obviously see that there's a lot more abundance in the effluent samples. And of course, this is sort of an average of various types of water quality from these wastewater treatment plants. And of course, they're a lot larger or a lot more profuse than the gene levels that we see in the freshwater samples. And if we take these and extrapolate to how much or how many antibiotic resistance genes are entering in copies per square meter, you can see that there's a huge amount of these genes that are actually being or that are infiltrating into these soils through these treated wastewater systems. And of course, these are real life examples. So it's quite worrying. And of course, the next step was to look in the soil and look at the crops and see what's going on there. And what we could actually see, and this is sort of a graphical summary of these measurements. And so we can see all of the soil here, and these are the crops that are being grown in these soils, and these are the actual crops. And what we can see in most of the situations that we analyzed, most of the crops and the soil, the gene levels were either detectable but not quantifiable or below detection limits. So our conclusion, and based on a previous experiment that we did that I didn't talk about, was that soil is an ecological barrier that impedes dissemination of antibiotic resistant bacteria and antibiotic resistance genes. So we have these ecological barriers, this resilience that's preventing colonization by these bacteria, and apparently we're not getting very much horizontal gene transfer either because we would have seen them. However, we thought of taking it a step further, and these are a number of experiments that were done by first Tamar Kozer, who was a master's student in the lab, and Chedi Lal Gupta, who was postdoc, and Roberto. And we did these microcosm experiments. I won't go into too many details, but first of all, we had these two very, very multi-drug resistant E. coli that were isolated from wastewater. And because they were so resistant, we could develop a media with different different combinations of antibiotics and show that we were able to isolate these bacteria without getting any soil bacteria that weren't amended specifically by us. And we added different concentrations and different soil types. I won't go into all the details, but what we can see very nicely is supporting our hypothesis of this resilience is when we add the bacteria to the soil, and this was a one-time spiking and then irrigating these microcosms with water without the bacteria, and we can see that their abundance went down at various rates until by 28 days we could not detect these bacteria anymore. And somebody would conclude that they're not there, but then Tamar did this very nice experiment where she took the soils here after 28 days, put them into rich media and then plated them, and lo and behold, with the exception of this microcosm over here where they disappeared after 8 days, we saw regrowth of these bacteria. And this is something that's very, very important because a lot of times, whether it's culture-based or molecular techniques, we say they're not there, and it's not that they're not there, they're just below limits of detection or limits of quantification. So we took this a step further, and I mentioned that we had some sites where we have tandem irrigation with treated wastewater and freshwater, and we had this avocado orchard here and this almond orchard here. These are both northern Israel, and then we had this very intricate lysometer system that was at Volcani, where we had lysometers irrigated with either freshwater or treated wastewater. And I won't go into all of the details here. It's been published. We did culture-based and molecular and metagenomic and quantitative PCR analyses looking at the freshwater and the treated wastewater. I'll just get to the point here. First of all, when we look at the microbial communities, these are the original treated wastewater samples. So these are the 3 different types of treated wastewater from the 3 sites. And these are the soil samples where originally we looked at 2 replicates. So we have the orchard 2 freshwater and treated wastewater. They're almost identical when we look at the soil microbiome. Sorry, this is the microbiome based on 16S amplicon sequencing. The same with the other orchard. In the lysometers, again, the freshwater and treated wastewater are slightly different when we look at the microbial communities, but then we do this enrichment, similar to what Tamar did before. So when we enrich the first orchard, we start to see between 3 replicates differentiation between the treated wastewater and freshwater soils. When we look at the second, again, see a similar pattern where again, there's a statistically significant difference between the enriched freshwater and treated wastewater. And we look at the lysometers. This is really, really impressive where we see this complete kind of separation between the 2. And again, we almost don't see a difference when we look at the original soil samples. And this is what we've sort of described as under the radar. And we know in microbial ecology, we always say that we use molecular techniques because culture techniques are very biased and they cause kind of induction of copiotrophic bacteria that grow very, very rapidly. But we're sort of trying to claim the opposite, where we want to try to do an epidemiological assessment. We feel that the copiotrophs are the ones that matter the most. And by looking at total bacteria using, culture independent methods, we're really getting a non-biased kind of portrayal of what's there, but we may be missing those under the radar copiotrophs that are very, very important from an epidemiological perspective. And so when we do these enrichments, especially, and I forgot to mention that the treated wastewater that was used in the lysometer experiments was very, very low quality secondary effluent versus the two others where we had high quality. So we see that when we use low quality water in a lysometer experiment, which might be more sensitive to perturbations than in field experiments, of course, we see this strong under the radar effect. And again, we feel that these enrichments are very, very important. And we see this in the lysometers also when we look at the total amount of antibiotic resistant genes. Again, in the orchards, we don't see it. This is after enrichment. And we also see it in the specific bacteria harboring antibiotic resistance genes. When we look in the lysometers, almost all the antibiotic resistance harboring genes that are enriched in the treated wastewater enrichments are Enterobacteriaceae, whereas in the freshwater enrichments, the difference between these two are here we see primarily gram-negative Enterobacteriaceae, and here we see mostly gram-positive bacilli. And again, I won't go into all the details. We see a similar effect when we do culture-based analysis of ESBL producing and carbapenem resistant bacteria. And I think this has a lot of importance for trying to understand the clinical potential of these kind of field experiments. I'll just shortly touch upon work that's being done now by Chagai Davidovich, who is a PhD student in the lab. His eyes are covered, not intentionally. And we're now working on this lysometer experiment that's within the premises of a wastewater treatment facility. How much time do I have left, Shimon? 2 minutes. 2 minutes. Okay, so I'll try to go very, very quickly. And again, here we created these lysometers with freshwater irrigation, treated wastewater irrigation, and then lysometers that were amended with poultry manure. And we took this a step further. First of all, instead of using LB, we used a media that's much more kind of indicative of the gut microbiome. We know that the gut has both aerobic and anaerobic zones, so we did enrichment with both. And this is kind of a work in progress, but I'll maybe jump forward a little bit. Okay. So again, when we look at the original, I forgot to mention, sorry, that we're looking at rhizosphere and phyllosphere now. So roots and leaves as opposed to soil, what we saw before. When we look at the original rhizosphere samples of the freshwater, treated wastewater irrigated, and the manure amended, we see that they all cluster together. Whereas when we enrich, so we have the enriched freshwater irrigated, enriched manure, okay? But then the anoxic and aerobic enriched treated wastewater are completely different. So again, we see this under the radar effect, which is very, very dominant. I'm going to jump directly to here. So this is analysis done by DSEC2, which basically shows you the differentially specific phyla in the different treatments. So we have the freshwater irrigated, manure amended, and the treated wastewater irrigated. This is the direct analysis. So without the enrichment, and we see that there are a lot of different types of bacteria, like the nitrosomonas, so ammonia oxidizers. But most of these enriched bacteria that we see when we do the direct analysis are environmentally relevant bacteria. On the other side, though, when we do the anoxic and oxic enriched enrichment and we look at who the dominant bacteria are, again, similar to what we saw in Roberto's experiment, we see that the more dominant families in the freshwater irrigated samples are the Bacilliaceae, so these gram-positive spore formers, whereas when we look at the treated wastewater irrigated enriched samples in the rhizosphere, we see Aeromonas, we see Enterococcus, we see Clostridia, and we see Enterobacteria. So again, these are more pathogen-related strains, and we have this under the radar effect where these copiotrophs are revealed. So to summarize, the limits of detection need to be considered. The fact that a particular antibiotic-resistant bacteria or gene is not identified does not mean that it's not present. Okay, and everything is not everywhere. Okay, so when we do these enrichments, we actually see this under the radar effect. And it can potentially let us identify anthropogenic impacts that aren't revealed using direct analyses. And I think this is very, very important. And finally, within a One Health perspective, targeting under the radar microbiomes and resistomes can be very, very important for risk assessment models. So just to really summarize to say where we're going from here, and after 10 years, you start saying, okay, what do we do with all this data? So we're trying to look at two directions. One is to try to use these enrichment types of platforms for quantitative microbial risk assessment. So how can I actually determine what the risks are of manure amendment, of treated wastewater irrigation within the context of human health? The other, and we heard a lot of talks about interactions with engineers. So this is a project that's just started with Sefi Warnick, who's from our agricultural engineering department, is to try to use bioelectronic-based devices to identify antibiotic resistance genes and try to get early detection. And if we can develop these devices, it allows us to get a lot more data, analyze a lot more points in time and in space, and hopefully be able to make better predictions regarding risk assessment. With that, thank all of the people who are involved and collaborators. And thanks a lot.
I have one question, short question, or we will leave and we'll talk to him on the way. So, because we are moving now to the Evans Auditorium. I hope you're moving on. Thank you very much for all speakers.